There are many variations on this basic protocol that I have shown, and I will talk about two of these variations. So in the CITESeq protocol, you are going to sequence the RNA as I have explained previously, but on top of the RNA, you're also going to estimate the abundance of cell surface proteins on your cells. And for this, you are going to use an antibody that targets a specific cell surface protein. And you're not going to use one single antibody, but you're going to use a whole panel of antibodies, typically 200 to 300 different antibodies that all target one specific cell surface protein on your cell. These antibodies are linked to a piece of DNA that contains a poly-A tail, a part that will bind to the sequencing primer and to the primer that is used for PCR amplification. And here in the middle, it will contain a barcode, a sequence of DNA that is unique for every antibody in the panel. Every cell surface protein, every antibody will have its own unique sequence here. So what you do is you allow your antibodies to interact with the cell surface proteins. You inject the cells together with the antibodies in the water droplets. You lyse the cells. So messenger RNA is released. Antibodies are released in the water droplet. And because all of them have a poly-A tail, they will bind to the sequences on the surface of the beads. They will be reverse transcribed, they will be amplified by PCR, they will be released, and they will all together will be sequenced using Illumina sequencing. So what you get at the end is the same data as before, so counts that reflect the expression levels of your transcripts in each cell, but also on top of that, you are going to sequence here the pieces of DNA that were linked to the antibodies that interacted with that cell. So, and that will give you a very good idea about the abundance of different cell surface proteins on the surface of the cell. What is the beauty of this system? The beauty of this system is that you measure both molecules, the RNA and the cell surface proteins, on exactly the same cells.